Alright, so we talked about eukaryotic cell division, was basically mitosis. Prokaryotic cells don't really go through mitosis. They have a different setup. For one thing, remember there's no nucleus, so there is no mitosis division of the nucleus. Also, rather than having multiple chromosomes, remember we have 46 different chromosomes in every cell, they don't. They have one circular chromosome. Still double-stranded DNA, and it's not in a nucleus, it's in an area called the nucleoid. And typically it's attached to the cell membrane. So when prokaryotic cells divide, it's actually called binary fission, which just means that the DNA makes an exact copy of itself and they split in half. It's very similar to mitosis, except there's not going to be any spindle fibers, nothing like that. It just, there's an extra circular copy and it divides. I think I have a little animation of it here, but I don't know if it'll play. There we go. So this is the start. There's your DNA. It makes a copy of itself. And then the DNA, the two strands separate, and you get a little cross wall formation, meaning basically just that the cell splits. And so this is how bacteria would make copies of themselves. So that's it. Really, the DNA, there's a lot of it, so it doesn't look like a perfect little circle, but if you were to stretch this out, it would be circular. It would be connected at both ends. There's a little origin of replication. It copies first, and then the rest of it copies, and there you go, two cells. And they're going to be clones of each other. This is a diagram of E. coli actually going through binary fission. This can be a positive and a negative thing. Obviously, if this particular E. coli has a genetic combination that allows it to survive in certain conditions, then that means that every time it clones itself, all its little clone offspring will also be able to survive under those same conditions. However, if we were to find, for example, an antibiotic that harms one of them, it's going to harm all of them because, again, they're all identical to each other. So sometimes there's a drawback to asexual reproduction and making identical copies of yourself. Okay, this is a little bit about control of the cell cycle. So there were two experiments that were done. If you look at the one on the left, they took a cell that was in S and they fused it with a cell that was in G1. And what they discovered was that the cell in G1 immediately entered S. And so, remember, that means the synthesis of DNA. And they did a second experiment where they took a cell again in G1 and fused it with a cell that was in mitosis. And as soon as those two were connected, the cell in G1 immediately started to undergo mitosis, even though that cell had never even been through S. You'll notice it doesn't even have an X-shaped chromosome, so technically it can't even properly go through mitosis. The idea is that they discovered there were some kind of chemical signals in the cytoplasm of a dividing cell or a cell in S that stimulate the chromosomes to enter the next phase. And so that's what this experiment was showing. And so they discovered that what it was is that there were these checkpoints during the cell cycle, and these proteins called cyclins were involved. And these enzymes would be produced, cyclin-dependent kinases, or CDKs, and when those enzymes were produced and bound to the cyclins, that's what stimulated the cell to go into the next phase. So if the cell makes, for example, too much of that enzyme, that cell may reproduce too much. And if the cell doesn't make enough, it may not reproduce at all. So for example, brain cells that are never going to divide again, obviously their cell cycles are controlled because they're not making these CDKs. That's what shuts the cell down and prevents it from going into S and going into M. And that's why that cell would then go into G0. Brain cells do this, muscle cells do this. Fat cells, any kind of cell, liver cells that are not planning on dividing anymore, it's because of these factors, the lack of certain factors that basically tells them to stay in G0 and not proceed. Again, some cells can re-enter it. If your liver is damaged, a lot of times your liver can repair itself. Somebody could actually donate a portion of their liver, be a living donor, as like donating a kidney. You could donate a part of your liver, and the liver would actually survive and regrow and heal itself. So this is sort of showing you the checkpoints. There's G1. If this G1 checkpoint, if there's no protein kinase interaction here, this is where it would just go into G0. Otherwise, it would go into S and then get to a G2 checkpoint and go into mitosis. So that's sort of how it's controlled. We're not going to get into a huge amount of detail about it, but what cancer really is a lack of control. What should happen is that these growth factors, CDKs and kinases, etc., they are supposed to send signals not to divide under certain circumstances. For example, if it starts to get crowded, if cells are touching each other, they should not divide. They basically send a signal saying, okay, don't divide, we're out of space. Or if a cell breaks off and it's not attached to any tissue, again, it should send a signal saying, don't divide, you're not attached to anything. But what happens in a cancer cell is that there's some kind of damage to the DNA so that those cyclins and kinases are produced when they're not supposed to be produced. There's nothing stopping the cell cycle. The cell cycle just keeps going. So the cells just keep making lots and lots of copies of each of themselves. Also, certain cells normally only divide a certain number of times. They don't just keep dividing forever. And so again, cancer cells we say that they're immortal. They just keep dividing over and over and over and over again. They don't get to a point where they stop. And so we think that something's wrong, obviously, with the cell cycle, and that goes back to their DNA, certain portions of the DNA controlling the cell cycle. This is showing an example. This is anchorage. The cells are anchored to this petri dish, and then what happens is once they get crowded, they're inhibited. If you take some cells out, then they will reproduce and refill that gap. That's sort of like when you get a cut and your cells are stimulated to divide to then form a scab. But what's going to happen in cancer cells, if you look on the right side here, is that there's some kind of a problem that the cell cycle is not shutting down. And so they just keep reproducing, and that's how you end up with what would be a tumor. So when a normal cell becomes cancerous, it's called transformation. And that means that basically the cell cycle has been somehow damaged. The DNA has been damaged so that the cell is not in check anymore. And your immune system can kill these cells, but if they're dividing out of control, a lot of times the immune system can't keep up with them. And so this is what a tumor is. It's just a mass of abnormal cells that just keep dividing. If they stay in one place, if you've ever heard of benign versus malignant, a benign tumor is basically a lump that stays in an original area and it's not really invading any tissues around it. It's not causing any problems. It's not stopping the organs around it from working, things like that. Malignant tumors means that it's growing enough that it's starting to invade the tissues around it. It's stopping those cells from doing their job, because any cell that's cancerous is not doing whatever its normal job is supposed to be. It's just dividing over and over again. And metastasis, or if a cell metastasizes, that means that the cancer cell breaks off. And if that happens, it basically can then float around to other parts of the body and attach somewhere, and then you'd get another tumor. So this is a diagram of a breast tumor showing actually the one on the right is liver, but it's showing the tumors. This is just big groups of cells. You'll notice that the big groups of cells aren't doing what they're supposed to be. Normal cells have a completely different look. These cells are just constantly dividing, and this is where then it could become invasive. So Hela cells and CHO cells, you're not going to be tested on them, but basically there was a lady named Henrietta Lacks back in, I want to say the 60s. She had ovarian cancer. They took some cells from her, and they're called Hela cells for her name, Henrietta Lacks. And they're still alive today, basically, in the lab. Labs can order Hela cells to do all kinds of research on, to try to figure out, how cancer spread or how to control it. But the cells originated from this lady, and then they've just kept them alive because they just keep reproducing. Since cancer cells are immortal, they just keep reproducing forever. Another kind of cell that they use are called CHO cells, which stands for Chinese hamster ovary cells, and they're another cell that are used in labs to study cancer. You can actually read about Henrietta Lacks. Her family apparently wasn't even aware that the hospital had kept her cells and that her cells are still being used today. And I don't know if the family ever got financial gain from that, but there is a story about it. And that's it. That's pretty much it as far as asexual reproduction goes. Asexual reproduction, mitosis would be asexual reproduction if it's a single-celled organism splitting in two. Then that would be asexual reproduction. It's a single parent passing on the genes. A group of individuals that are all genetically identical, those are basically all clones. Sexual reproduction, which is our next section, has to do with two separate parents or two different cells combining their chromosomes, and so now you're getting a new combination because you're not just passing on an exact copy from one cell, you're basically getting a combination of two cells, chromosomes, coming together. And so the way that that works, the way that we prepare for that is meiosis as opposed to mitosis, and that's what the next section is going to be about.